over here. I know whenever I say Yersinia, all of you are only thinking of Yersinia pestis, plague. No, that's not the right way of approaching it. Yes, you are thinking of Yersinia pestis, which causes plague, but do you know Yersinia pestis doesn't come under this Enterobacteriaceae family? I am just reading it over here for completion, but ideally Yersinia pestis is causing plague, but doesn't come under the Enterobacteriaceae family. It's a miscellaneous bacteria. What comes under the Enterobacteriaceae family is known as Yersiniosis. These are Yersinia enterocolitica and Yersinia pseudotuberculosis. So you will say fine. The tribe Yersinia has Yersinia pestis, Yersinia enterocolitica and Yersinia pseudotuberculosis, out of which pestis causes plague but is not a part of this family. Enterocolitica, pseudotuberculosis, together known as Yersiniosis, are a part of this family. So if I have to do their differences, first thing that I will do is their ability to produce urease. So remember, these Yersinias of mine, which are Yersiniosis, produce urease. So remember the years, although it's not very important, that's why I did not put it much in the punch kiss mnemonic. But over here you need to know Yersiniosis is going to cause ureases. Okay, that is how I have learned it. Yersiniosis wala organisms are going to produce ureases. After that, please note, we have to talk about their motility. So the Yersinia pestis though is non-motile. So neither is it showing motility nor is it showing urease. But this Yersinia enterocolitica and pseudotuberculosis, they are. And they have a little weird thing. See, they are motile at what? They are motile at 22 degree. At 22 degree, they are motile. But they are not motile at 37 degrees centigrade. What is this kind of motility known as? This is referred to as the differential motility. And now can you recall for me, if I have to make a table over here, the differential motility in which you were seeing that you can see motility at 22-25 degrees centigrade, but you don't see motility at 35 or 37 degrees centigrade. So did I tell you there is one organism which is tully all the time, drunk organism, keeps tumbling and falling? Listeria. We had studied that Listeria is also tully and tumbling, and that tully and tumbling organism could drink in his 20s but could not drink in his 30s because of age. That is how we had learnt it. Can drink in the 20s but cannot drink in the 30s. Same way, Yersiniosis is also like that. Can have motility, roam around in 20s but cannot roam around in the 30s. So remember, these are the two organisms which are known to cause this. Yersiniosis and Listeria show differential motility. Now, obviously, for the time being, first I'll quickly finish off these two Yersiniosis, that is Yersinia enterocolitica and pseudotuberculosis. If you see, they are associated with everything to do with GI tract. So there is gastroenteritis, there is mesenteric lymph node involvement, there can be terminal ileitis. In fact, it can many a times mimic appendicitis because it comes to you with pain in that region. Pain in the appendix region is going to mimic appendicitis. So many a times this is also known as pseudoappendicitis. Please note, Yersiniosis is many times known as pseudoappendicitis. So basically I realized primary condition is going to be the GI tract, gastroenteritis, mesenteric lymph node involvement, ileum involvement, appendix involvement, and in adults it can also cause some other organ involvements, right? So now we come down to how are you going to do the diagnosis of this Yersiniosis. Means I'm talking about Yersinia enterocolitica, Yersinia pseudotuberculosis. I'm not focusing on Yersinia pestis for the time being. So remember, there is going to be a McConkey agar, and I had studied that Yersinia and all of them are non-lactose fermenters, but this Yersinia enterocolitica can sometimes show lactose fermentation. Next, what is the mobility? This I've already done. It can move around in its 20-25 degree, but it cannot move at the 37 degree. This information I have already done. Same goes for its temperature. This follows something called cold enrichment. What is cold enrichment? That ideally it will grow somewhere around 25 degrees. But if you incubate it at a very cold temperature, it will get enriched. It will start growing easier. The same phenomena was also seen in Listeria. So now you can see that there are a lot of similarities that you have between Listeria, or I will write it Listeriosis, and Yersiniosis. Okay, that becomes rhyming for us. Listeriosis and Yersiniosis, both of them show you a phenomena that is known as cold enrichment, and both of them are going to show you a phenomena known as differential motility. Means they can move in the 20s, they cannot move in the 30s, right? And cold enrichment means that at cold temperature of 4 degrees, they are going to be grown easier, they are going to be isolated easier, so they are coldly enriched. This is the main point, even important for Yersiniosis. And now I can also finish off Yersinia pestis telling you Yersinia pestis does not belong to the Enterobacteriaceae family. For completion, I'm taking it up over here. And Yersinia pestis is the one that is going to cause plague. So what are the 4 endemic foci for plague? Tamil Nadu, Maharashtra, Himachal and Uttaranchal. So if you're from any of these places, just for your general knowledge, I'm telling you now that they're going to ask. So now my concern is that how does plague happen? I mean, what are the vector and organism and all? Then what are the virulence factors? What is going to be the signs and symptoms? The diagnosis, the treatment, the vaccine, as usual. So first let me start off with what kind of an organism or what kind of a disease is plague. It's a zoonotic disease. Zoonotic means it's coming from the animals to the humans. Okay, so who is the reservoir? We know this is rat wala disease, and rat, say who brings it to us? Of course, that is going to be Xenopsilla, means we have rat flea. So there is going to be a rat, and via a rat flea, it is going to be coming into us. So that is Xenopsilla, Cheopsis or Astia. Different species are in North India, South India, right? So how does it spread? I hope you know it is basically by the bite of a rat flea. I'll tell you the entire organism's life cycle, how the rat flea is going to take it from the rat and how it's going to give it to the humans. But in PSM, you do the rat flea index. What is rat flea index? So you're being territorial, that how many fleas are present for how many rats. So number of fleas divided by the number of rats, that is going to be the flea index. So if you want to find out the flea index, keep flea in the numerator, number of fleas divided by number of rats. So simple, the reservoir is the rat, from there the vector is a rat flea, and it's going to come and give it to humans, so it becomes a zoonotic disease. So what is going to be the entire life cycle? Firstly, we say that okay, there is a rat that is in this story. So the rat flea is going to first go and bite this. So it is going to get infected from the rat. It's going to take the infection from the rat. What will happen? In the stomach of this rat flea, now there is going to be a multiplication. The plague is going to multiply. That is why we call it what? Go to your parasitology lectures. I have taught you that the type of vectors are propagative vectors. So this happens to be a propagative vector. Propagative means that this is propagative. We had studied that, see, vectors are 2 things. Either the organism is going to develop in them, or the organism is going to multiply in them. So propagative are those PR in which there is going to be replication. So can I say rat flea is a kind of a propagative kind of a vector in which there will be multiple, I can see multiple plague organisms formed. And now what do? This was happening in the stomach. From the stomach, you can't go and infect someone. So it will come back and block this salivary gland area. In the flea, I am becoming a flea for the time being. So for the time being, all this organism is multiplying in my stomach, and after some time, it's going to come back and block the proventriculus of the flea, so that next time when it bites, it's going to spit out everything. Next time when it bites, it's going to regurgitate everything in the blood meal. So imagine, from the time it took it from the rat, number 1, to the time the proventriculus is blocked, that is the extrinsic incubation period, which is approximately 2 weeks. Repeating once again. From the time, and I'm going to repeat this entire cycle once again for all of you to be very clear. What happens? Firstly, there is a rat, and that rat, from this rat, the rat flea has taken this entire bacteria. It has gone into the stomach, and in the stomach it has multiplied. So it is a propagative vector. After that, it is going to come and block the proventriculus. So from the infection to blocking of the proventriculus, it is going to be the extrinsic incubation period, means not in humans, bahar wala incubation period. And finally, when it's going to take a blood meal in the humans, it's going to come and give it. So it is zoonotic infection. There are lots of virulence factors. If you're one of the inquisitive ones and you can learn so much, please do. But what you have to learn, that is, there is an envelope antigen called F1, because later on the vaccines are also made out of it. In fact, they say over here it's nicely written as a protein envelope antigen. This is also known as a protein capsule. This is also known as a protein capsule, and that is a difference, because I have always learned that capsules are made up of polysaccharide, and now I am reading over here that capsule can also be made up of protein. Yeah, this is not very commonly known by students. So please note that protein wala capsule is the F1 capsule, and protein hai, so it is plasmid coded and it is heat labile. I will be using this in the vaccine in a while. Other than that, it has VW antigens, coagulase, fibrinolysins, it has toxins. Again, as I said, This is there in every bacteria. If you can learn it, the enzymes, the toxins, very good. But F1 protein capsule or envelope, you have to know. So now tell me, if there is an animal involved, there is a flea involved, and there's a human involved. So what are the cycles of plague that we have? One is an urban cycle. Urban cycle means that this is going to happen between the rodents and the humans. Whereas there is a wild sylvatic cycle. So देखो जो urban वाला है means where people are living, domestic cycle. So we are humans and there are rats and the cycle is happening between us. But if it is a wild or a sylvatic, this is only between the wild rodents. In this, there is no human involved. So two cycles. One is urban between human and rodents. One is wildlife cycle that is between rodent and rodent. And what are the types of plague that you have? Bubonic plague. Why did I count like this? Bubonic has 7 alphabets. So the incubation period is roughly going to be 2 to 7 days. If it enters septicemic, anything can enter septicemia. That will also have 2 to 7 days. However, if it has a respiratory involvement, that is going to happen very quickly in 1 to 3 days. So repeating, the bubonic 7 alphabets, there's going to be 7 days. Respiratory wala plague, very, very quick. And anything going to septicemia, again 7 days. So what is the bubonic plague? The bubonic plague basically means that there is a painful lymph, bubos, means bubos is always lymph node. Painful lymph node is bubos, and fever to of course hoga. Pneumonia, guys, come on, pneumonia we know is going to be respiratory symptoms, and this is associated with a bioterrorism agent. Coming to septicemia. Septicemia means ultimately even bubonic plague can go to septicemia, pneumonic plague can go to septicemia. Septicemia means the patient is going to have sepsis. Also, this is known as black death. Why is it called black death? Because patient can have sepsis, shock, DIC. There can be multiple organ failures. DIC with shock, with multiple organ failure. That is why, guys, plague is also known as black death. Hai na? So repeating, there's a bubonic plague in which there are painful buboes or lymph nodes. Pneumonic plague, which is of course respiratory. Anything can go into septicemic plague, that is black death. Bubonic and septicemic 7 days, pneumonic is 1 to 3 days. So how do you do the diagnosis? Firstly, what sample will you take up? Depend. If it is a bubonic plague, you will take sample from the buboes. Means you're going to take the bubo sample. If it is a respiratory symptom, then you will take sputum. If it is some other multi-organ involvement, you will take as per that. So you will take the, the final sample depending on the site. And what does it show you? Once again, it shows you the famous safety pin appearance. Yes, microscopically, you see the safety pin appearance. Another name for safety pin appearance, I told you, it looks like this. This means it is a bipolar staining. Safety pin appearance refers to bipolar staining, and I've got you all the organisms in microbiology that show you that. So for that, all of you will have to make a safety pin, and I have no mnemonic because I think by now you are sick of mnemonics. That ABCD has also stopped working. Hai na? So now how will you learn it? So let me see. From one end of the safety pin, can I make a Y? Yes, Yersinia pestis. Can I also from the bottom make a V? So that is going to be Vibrio parahemolyticus. Okay. From here, can I end up making a D? So Haemophilus ducrei. From the D, can I make a B? All the Burkholderia malai, Burkholderia pseudomallei. And finally, we have that I can bisect it and make a K. And that K means Klebsiella granulomatis. Remember I told you the Donovan bodies a while ago? Exactly. So do you want to do this? A lot of drawing that I've done. So pehle in the exam, make a safety pin. If you make a V out of it, it is Vibrio parahemolyticus. If you make a Y out of it, it is Yersinia pestis or plague. If you make a D out of it, Haemophilus ducrei. You make a B out of it, Burkholderia malai, pseudomallei. And you make a K out of it, Klebsiella granulomatis. Bus fill all the lines, practice this once on your own and you are sorted. These all show you the safety pin appearance. But now the question comes is, what is the stain used for knowing this safety pin appearance? Most importantly is the Vasen or the new methylene blue, also the right or the Giemsa stain. If you have to mark one, you have to mark the new methylene blue or the Vasen stain. So remember, Vasen or new methylene blue, also right and Giemsa can be used. What else? What are you going to use for the culture? So let me show you two pictures. Firstly, you can see that there is something known as the CIN agar. And it's a long name. Please don't get into it. Don't even Google it. It's so confusing, so lengthy. You don't have to know. For Yersinia pestis, you have the CIN agar, and that is going to show you the very famous bullseye colonies. You can see the bullseye beautifully over here. Apart from that, you can also see the ghee broth. There is something known as a ghee broth, and from top of the ghee broth, do you see something is falling down? So that is referred to as the stalactile growth. So remember, selective media was CI and agar, that is going to show you the bullseye colony. Whereas if they ask you what is the ghee broth, that is going to show you the stalactile growth. Stalactile growth means something is hanging, dropping from the surface. So stalactile growth and bullseye, repeating those, this is referred to as the stalactile growth. It is hanging from the top, and bullseye of course looks like a bullseye. These are the two main things. After that, you can definitely know the others. Like any other thing, you can grow it at a culture of 27 degrees. 37 पे it might develop an envelope. Everything I'm realizing, it is very comfortably, whether it was Yersinosis also, 20s mein everything is going fine. 30s mein jaake it has developed an envelope. Okay. On blood agar or chocolate agar, this can give you a dark brown color colony because of the absorption of hemin. So I realized that Yersinia pestis is not only going to be a black death, means plague, it is also going to show you a black blood agar. The black blood, so black death may be bloody death ho raha tha, and black blood agar or brown blood agar is also noted. But these two, please don't forget, it shows you the stalactile growth and it shows you the bullseye. So how I have learnt it is, for example, this is because of rats na. If you want to kill a rat, thoda sa imagination, I tried searching for pictures, I couldn't get. If you want to kill a rat, what are you going to do? You have those rat traps na. So how do they show you in the cartoons? How have they killed the rat? They pick a rat with the tail. They always pick a rat with the tail like this, and they show you that the rat has been hanging rat. So stalactile, something hanging. They always show you a hanging rat, and if they want to catch the rat, they put a cheese or something. That's how they show you for mice and all. So they put a cheese or Tom and Jerry wala, and they trap it, matlab bullseye. Bullseye for catching the rat, and hanging rat, that is, I've caught it, stalactile growth. So stalactile growth and bullseye is for the rat wala disease, and that is going to be your Yersinia pestis. The diagnosis is not over as yet. You've only done the culture. What about the biochemical reaction? So we have realized that the glucose, mannitol, maltose, basically the sugars are going to be fermented only with acid production. No gas production. Catalase positivity, yes. Oxidase negativity, but that is for everyone in this family, na? Enterobacteriaceae, catalase positive, oxidase negative. You will tell me IMVIC testing. Is pure mein IMVIC is important. So tell me something, guys. I gave you a mnemonic called I am shy, and I am shy said that when you are talking about Shigella and when you are talking about Yersinia, you are gonna be interested only in the methyl red test. So minus plus, minus minus. Only the methyl red testing is there in Shigella and Yersinia. So I am shy. So how are you going to do the serology? Ab now go back, think what was the antigen? Ma'am was focusing a lot on the F1 antigen. So against the F1 antigen, if I get antibodies at a titer of more than 1:128, that is definitely going to be very important. So repeating, antibody against F1 and more than 1:128, that is the significant titer. And what about rats? So please remember, if you have rats, first and foremost, before you start isolating this organism from the rats, make sure you dip it in 3% Lysol so that all the infected flea and everything has been, destroyed. So whenever you're dealing with any animal study for plague, always dip it in 3% Lysol. Okay. Now finally, vaccine. Abhi bhi common sense hai that the vaccine had to be prepared from. So there is a recombinant F1V vaccine. Vaccine is also prepared from F1 antigen. There is something known as the Sochi's vaccine. Now please remember, Sochi's vaccine is going to be 1, 2, 3, 4, 5. It provides the protection for 6 months, or you can learn it as Sochi's also. That will make it better. So if you learn the Sochi's vaccine 1, 2, 3, 4, 5, 6, 7, तो 6 or 7 months तक का protection है, and the protection is only against those 7 alphabet वाला bubonic. The protection is only against the 7 alphabets wala bubonic plague. It is a formalin-killed vaccine. So Sochi's modification of Hafkeen vaccine. Remember, Sochi's modification of the Hafkeen's vaccine is Sochi 7 alphabet, protection against bubonic plague 7 alphabets, protection approximately for 6 to 7 months. But if you ask me what is the treatment is going to be streptomycin. This treatment is going to be streptomycin, and if there is involvement of brain, or there is involvement of lungs, or there is involvement of heart, then you can add the fluoroquinolones. So you can add the